Good afternoon, and thank you so much for joining our first virtual 9th Annual Forensic Science Symposium Lunch and Learn poster session hosted by the Global Forensic and Justice Center and sponsored by Foster and Freeman and Promega. We are recording today's sessions and we'll let you know when it's available on the GFJC's YouTube page. You can find it at go.fiu.edu/GFJC YouTube. Please use the chat function feature to ask your questions. Any unanswered questions due to time constraints will be provided in a written format shortly after the presentations this week. Also, you can make this presentation full screen by clicking on the 4 arrows icon in the top right corner. To return, you can click the same icon again. Now, with those housekeeping items out of the way, I'd like to present today's presenter, FIU grad student Nicole Tejero, with her presentation. Nicole, please take it away.
Thank you so much, Michelle. My name is Nicole Fernandez Tejero, and today I will be presenting my research on the development of epigenetic methylation markers for skin and sweat. I work in Dr. McCord's lab on the epigenetic lab, or part of the lab, and this I did with Quentin Gauthier, Sohi Shah, and Hussein Al-Hamim. So you will ask why we want to do the development of epigenetic methylation markers for skin and sweat. Well, basically, the determination of tissue type is important when reconstructing a crime scene. However, at present, there is no specific methylation-based marker to distinguish touch DNA from other body fluids. Because of the lack of epigenetic markers capable to identify sweat and/or skin, we are exploring the genome to develop markers for skin and sweat. Typically, the DNA in such samples results from free DNA secreted in sweat and cellular material, resulting from skin cells. Work in our lab has successfully identified DNA methylation in various body fluids, but not in skin or sweat. Moreover, the development of a body fluid identification multiplex in our lab will greatly benefit from the inclusion of a skin sweat marker. Our goal is to develop methylation-based skin markers for their use in forensic analysis. In this study, we compare DNA obtained from skin with semen, saliva, blood, and vaginal epithelial. I will explain everything when moving forward with the poster. An Illumina methylation EPIC peak chip array was utilized to perform a genome-wide association study to determine suitable methylation markers for touch DNA. The results indicated 5 different genes for potential use for skin and sweat methylation markers. Those are WDR11, PON2, NAA16, MRPS15, and NHSL1. After this identification, we designed PCR primers to encompass these loci. Afterwards, we started with the sample collection. Next, we did the DNA extraction. We quantified and bisulfite converted using the Qiagen bisulfite kit, where, as you will know, unmethylated cytosines will be converted to uracils and then on the DNA template. Here, I used the high-resolution melt to analyze for different melting temperature. So what we do is that the different body fluids will have different melting temperature, or that's what we wanted to verify, and you will see that it was possible. So afterwards, you will see that these results demonstrate that high-resolution melt analysis may be a promising technology to identify the tissue source of a given DNA sample. So we started off with doing the GWAS, the genome-wide association study. So here, a series of human body fluids were screened for the presence of tissue-specific methylation of skin and sweat using the Infineon Human Methylation 450K bit chip, identifying 850,000 CpG sites throughout the genome. Samples of sweat, sperm, saliva, vaginal epithelia, and blood were screened, and as I said before, 5 CpG locations were identified. We created the markers for them, and then we have to do the bisulfite conversion and then create the primers. So in the materials and methods, what we do is that we actually collect the sample. We collect the sample by asking participants to do their routine workout, and then I just give them the swabs, and they swab their forehead when they have a little bit of when they have a little bit of relaxation from the sweat. So afterwards, I do the DNA extraction that basically, as you know, it functions to lyse the cells to release the DNA molecules and to separate the DNA molecules from other cellular materials. After the extraction, we quantify the DNA by using the qubit 4. Here you can see the qubit 4 fluorometer from Thermo Fisher, and for the DNA extraction, I'm sorry I didn't say, is the DNeasy blood and tissue kit from QIAGEN. After we do the DNA extraction and quantification, we will do the bisulfite conversion. And the bisulfite conversion is actually known as the gold standard for DNA methylation analysis, where unmethylated cytosine residues are converted into uracil by the bisulfite. Well, the methylated cytosines will remain unchanged. Therefore, bisulfite conversion treatments gives rise to different DNA sequences for methylated and unmethylated DNA. Methylated cytosines will remain unchanged under the bisulfite treatment. This step is key for DNA methylation studies because standard PCR amplification will not retain methylated sites on the DNA. So this is why we actually use the bisulfite conversion. In PCR amplification, uracil nucleotides will be recognized as thymines and then paired with an adenine. So after we do the bisulfite conversion using the EpiTech Fast Bisulfite Conversion DNA kit from QIAGEN also, we then do the high resolution melt. So basically, we will be used as screening. It will be used as a screening process to assess individual PCR amplicons based on differences in methylation content between bisulfite modified PCR amplicons and between PCR amplicons. Yeah. So the HRM is a real-time PCR method that utilizes an unlabeled primer pair for amplifications and includes an intercalating dye for amplification detection and melt analysis. So after we do the high resolution melt, we then do the data analysis. That this is what I will be showing you guys today. So as we can see in the figure A, so figure A is actually one of the bar plots that we got from the GWAS, or from the methylation epic beat chip array. So what we will get is like I hope you're seeing my cursor. You will be getting like a red and green type of signals. And this will be telling us like where are the methylated or unmethylated. So this is one of the 5 genes that we discovered, and here you can actually see that sweat has a different methylation from other body fluids. So this is what we wanted to see. I only added one because of space, but we can see the same thing on throughout the other body fluids. So after we saw the difference in body fluids, we actually the difference from sweat and other body fluids, we decided to create the primers, as I said before, and now we started doing all the methodology, and we did the high resolution melt. So this is actually an example of the HRM graph. It will be plotted as the differential of fluorescence in function of temperature and sweat, will be the Y, and the X will be the temperature. So we wanted to see that the theoretical from the experimental, basically, we wanted to see that sweat will have a difference in methylation. I'm sorry, the difference in temperature, therefore a difference in methylation. So we can actually see here that sweat has a lower, specifically for pond 2, that's the one that I'm showing here, both of them. We can actually see that sweat is more to the left, therefore it has a more unmethylated trend. So finally, after I plotted this in a bar plot, we can see here 4 different genes, I'm sorry. So we can see here, for example, sweat, sperm, saliva, and vaginal epithelia, and blood. They are the same colors on all of them. If you forget, they're down here. So this one is pond 2, the same as all of these ones, and this one is MRPS 15. This one is WDR 11, and this one is NHSL1. As I said before, there were 5 body fluids there were 5 genes. 5 genes. I am only putting 4 because of space. But here you can actually see, for example, in WDR11 and NHSL1, you can actually see the difference in temperatures from the other ones. So this is a good preliminary results that we can actually keep on moving forward with this, and it also lets us know that we can actually identify skin/sweat from other body fluids. So in conclusions, we have performed a GWAS study using the Illumina methylation bit chip array to detect epigenetic biomarkers for skin and sweat, and 5 CpGs were identified as potential methylation markers for touch DNA. Preliminary results show that HRM is a promising technology to identify skin and sweat in forensic analysis. But my research will not stop here with HRM. I really like using HRM as a preliminary type just to see if it works. If it works here, I will hope that it will work on the other instrument that I'm going to be using, that it will be the pyrosequencer. And basically the pyrosequencer is when you use sequencing by synthesis, which involves a single strand of DNA and synthesizing it complementary strand enzymatically. This instrument, we have it on the lab. It's the PyroMark Q48. So that is one of my future studies, and hopefully I get results there. And finally, of course, perform validation studies to include degradation, sensitivity, and mixture resolution. So I still have a lot of work to do, but I think that these are very good preliminary results. My acknowledgements, I want to thank my TVBS fellowship, McKnight Fellowship, the National Institute of Justice, and of QIAGEN, and of course McCord's Laboratory for all their assistance. If you have any questions, please feel free to ask. We'll be here.
Wonderful job, Nicole. Thank you very much. And as Nicole stated, if you have any questions, feel free to put them in the chat session and we'll be asking them here. Nicole, I do want to get started on some questions. You said you still have some more work to do. What are your plans? Do you have anything else that you would like to explore on these preliminary results?
Yes, I want to add more. Actually, I want to add more body fluids. I would like to add even menstrual blood just to see if it differentiates from it. Because remember that from menstrual blood, you will have blood and, of course, a little bit of skin from there, you know. Besides that, as I said before, I want to use the Pyro sequencer because it will be more cost effective. It will be more fast, basically, and of course, like do the validation studies. I think those are my main goals this year.
So do you have about a cost analysis per test on what those would run if they were actually being used in a crime laboratory?
I'm sorry, can you repeat it?
Yes, of course. Do you know about how much the test would cost if this was being used um, the way that you have it set up right now in a
Mhm.
crime laboratory?
You know, I haven't actually done the math per se on the HRM. I haven't done the math. I'm sorry, Michelle. I will have to get back on that. But that's a very good question.
No, not a problem. We just had a lot of questions about cost over this webinar, so I just wanted to add that in for anyone who is asking. We do have a question. What are the general amounts of DNA present in the samples being tested?
So let me get my. It was like 1.2 nanograms specifically from sweat. We can get like from 1.2 when we can get one from 1.2 nanograms to like 7. And what is the point?
And how much sample is required? Okay, this may have answered it, but I just want to make sure because it is a slightly different worded question.
Mhm.
But how much sample is required for the HRM analysis?
How much sample? I think it was like I usually add like I'm using actually now 1.6 nanograms of body fluids for all of them. After I do the bisulfate conversion from there, I just get like 1.6 and add it to the master mix of the high resolution note.
Wonderful. And then we do have another question. First of all, they said good work, which I can agree with.
Thank you.
Could you tell us the rationale behind choosing the 5 CPGs site out of the 8,500,000 CPGs? I apologize. I'm a non-scientist reading these questions.
No, no
Um,
worries.
So if I say something incorrectly, feel free to correct me so that I am saying it correctly for the audience.
So let me see if I'm understanding correctly. They're asking why use those 5 genes from those 5 CPG sites from all of the CPG sites they're scanning off, right? The machine is scanning.
So if you will look at your chat, I sent you
Oh, okay.
the question wording because maybe it's better from a scientist looking at it to answer it more appropriately.
Okay. So specific, yeah, it's thank you, Michelle. So basically, this CPG site specifically, I think they're seeing my cursor, right?
Yes.
Okay, perfect. So what we do is that this machine will scan all of those CPG sites, and where they see a very good trend of differentiation, that will be that's the CPG site that you can see difference in methylation. So here you can actually see that sweat is different from all of the other body fluids, and that's what we want to see because we want to see differences, a very good difference from other body fluids. So here you can actually say if I give you this if I give this result to you, you would say, yeah, that's definitely sweat because it has a lower methylation than the other body fluids.
Okay, and another question for you is what is the advantage of using DNA methylation to determine body fluid as opposed to RNA to more directly measure the expression of these genes?
I think that DNA methylation is good because it's something new. I think it's faster, and it's not that RNA is not good. We're just scientists and we're just looking for more and more things where we can promote or we can help the scientific community here.
Wonderful. So these have been great questions so far. So please feel free to chat either Michelle, that is me, your today's moderator, or Nicole, the FIU grad student who is presenting her work. Thank you very much. Now, Nicole, to kind of go off on, to jump off the last question, have you done a comparison between this and the RNA, your methylation and the RNA?
So I haven't done a comparison because this is still very new. There are no methylation markers for skin and sweat. There are methylation markers for RNA. So this is why it's so important and so cool, I may say, because we can actually do a comparison afterwards, and that's a very good suggestion.
Wonderful.
Mhm.
So we have another question. How long of the time did you need to get the result? How long did it take you to get your result?
From the whole method, yeah, so I believe it will be from materials and method. So the DNA extraction, I think it will be like 1 day and maybe a little bit more, because I know I remember that one day I did all of it. I did a lot of samples in one day, started from DNA extraction to quantitation to isotope conversion. Like I did the whole process, and I just did the data analysis at home, and it was a whole day. But if you want to divide it, it will be like a little bit less, a little bit more. It will just be on how handy or how stressed you want to be in the lab, basically.
Sure. I hear everyone likes to be stressed, right? So how can DNA methylation be affected by the age of the contributor, like young versus the elderly?
So that's a very good question. Actually, my lab has studied methylation markers for age. I think I don't remember right now the results, but we can actually see different methylation markers, different methylation percentage from different ages. So you will have a gap of, let's say, methylation. A low methylation will be for a person that has 24 to 20 to 34 years old, something like that. You will have percentages of DNA of methylation for the different ages.
Great. And another,
So let me add something else on
sure,
top.
of course.
Here, I haven't studied specifically age. I've only been studying skin and sweat. So I have been, so people who go to the gym and whatnot. So here I have only been getting results from more of a specific age gap. So I can add another future work that will be including ages, like different ages and how the skin or methylation will change. I wouldn't expect to change a lot. I wouldn't expect to change, but yeah, that will be another future work.
These questions are just keeping your research busy. That's all. That's
Yeah,
all that's happening.
that's true.
Wonderful. So are you looking at any additional genes to improve the resolution between saliva, vaginal, and blood?
So as of now, no. I want to concentrate specifically on these five that have given us very good results and that has given us like light that it can be done. But of course, after I finish this specific work, we can actually see more. We can actually perform another GWAS and see if we get other CPG locations.
Okay. And what were some of the issues you encountered when performing this experiment? This person also wants to know what program was used for the data analysis for the melting temperatures.
Okay. So one of the issues was more on sample collection. I've stayed a lot of time in sample collection because it's not easy to get skin/sweat. Sweat is basically water. So yeah, I have to see how we got better results or better DNA from the different collections. So I saw that by swabbing the forehead after you did your exercise and then letting your forehead dry a little bit and then swabbing it there, it actually gave better DNA. So that's a good thing that I at least saw that. And afterwards, I think the DNA extraction was the hardest part. We tested with different QIAGEN kits, and I think this one was the best one of all of them. So the NEC blood and tissue kit from QIAGEN. We also used the easy one. We did this one. We did the organic extraction, and basically this one was the best one of them. And the other question was the data analysis, right? So is it this data analysis or this one?
They said specifically melting temperature.
Melting temperature. So it will be this one. So I just use Excel. After you do the HRM on your rotor gene, you will get like all the plots to get this curve, right? So after that, I just went to Excel and I saw which one was the highest peak. From there, I just plotted it, and on Excel, basically, I took the highest peak from all of them and just plotted it here with the melting temperature.
Okay. And another question from the extraction end, what were the limitations by using this method?
The high resolution melt.
The extraction, whatever your extraction method was,
Okay.
what were the limitations of using your extraction method?
So right now, this method, I don't think I have had any limitations. It's been more the search for a better DNA extraction, which gave us better results or better quantity of DNA. But really, I really like it a lot. It's very straightforward, the process for the DNAC blood and tissue kit. I don't take a lot of time doing the extraction, and really I feel like I enjoy doing the extraction with this kit. Like it's very, I don't know, it's very cool. I like it a lot. But limitations, no. Not with using this one specifically.
So they came back and clarified
Mhm.
the HRM
Oh, okay,
method.
okay.
Yes,
That's
yes,
what I
yes.
thought. That's what I thought. So again, I haven't encountered a lot of high resolution. I haven't encountered a lot of limitations with HRM. This is preliminary, so it's not that these are my final results. As I said before, my final result is my final goal is using the pyro sequencer, but I used the high resolution melt to actually see if we could see different trends in methylation. So some limitations, I'm trying to see, maybe doing the samples one by one. Yeah, maybe that. Like you have to prepare the samples one by one, but I think that will be all.
Okay, and we had a clarification question. Are the samples used here from the individuals are your individuals around the same age?
I would say yes. They will go from like 24 to like 30, 34. I think that was the age gap. So maybe like a 10-year age gap.
And then have you looked to see if there's any difference between stress sweat versus normal sweat?
No, I haven't. I'm still in this type of sweat, but those that is one of the things that I have in my in from my dissertation committee. They have uh, talked to me about doing that, and that's a very good recommendation.
And so your current is on the exercise sweat,
The
correct?
exercise,
Okay.
yeah. Hmm.
And are you expecting to see a difference? Do you have a hypothesis on that?
Do I expect to see a difference from stress sweat? And I think maybe yes, depending of I don't I haven't actually thought about that, but maybe yes, because those are different types of sweat, right?
Well, at least one deodorant commercial told me that it was. So,
Yeah.
and you only tested the forehead. You swabbed from the foreheads, correct?
Yeah. I
Okay.
swapped on the foreheads, and I think and I did another place more. It was the forehead and the hands, but I decided, and after I did the DNA extraction and the quantitation, we saw that we actually got more DNA from the skin in the forehead. Yeah.
So again, if anyone's just joining us here in Nicole Tejero's room, she is presenting a very wonderful research project. And please feel free to add your questions to the chat. If we do not get to them, because we do have a little less than 30 minutes left in today's poster presentation, I will be sending these questions over to Nicole to answer, and so they will be available in a written format. So your questions will be answered even if she doesn't get to them on the audio level. Would it make sense to ever do a comparison between the sweat collected from the forehead and or the hands to a different part of the body? Would you think that that would be the same results?
So it makes sense. I would like to do it just to see, but I think it will be the same result. The only thing that will change will be the DNA quantity. That's what I'm thinking, that it may be different.
Nicole, what made you ask this question to start this research?
So when we were doing preliminary literature search, we didn't encounter any methylation from for skin and sweat, and we think that that's one of the most important body fluids also. So our lab already did the markers for sperm, saliva, vaginal epithelial, and blood, and that's why we decided to add sweat and skin, because of course when you're in a crime scene, when you're the perpetrator, I would believe that you will sweat a lot, right? And that's why we wanted to add sweat and skin. Also, Quentin Gauthier, one of the co-authors here, he did he combined all of the body fluids in only in one uh, one assay. So we want to actually add this for the for that assay. He did a multiplex. I'm sorry, that's the word. He did a multiplex, and we would like to add sweat to that multiplex.
Do you have any idea what your validation studies will look like?
Yes, I will be doing degradation, sensitivity, and mixture resolution. Mixture resolution will be when you have more than one body fluid in the same sample. So for example, let's say I have skin and sweat and sperm. We will start seeing like what is the percentage, and see like how it will be, how much variability we will see. Also, degradation, well, of course, when the DNA starts to degrade, we will see until what point we will see, we will still be able to differentiate from other body fluids when the DNA is degraded. And the sensitivity is that as how low can you go, basically, how low can you still be detecting your body fluid from the other body fluids from low collection of DNA.
So, ladies and gentlemen, we have about 20 minutes left in today's poster session. We thank you so much for joining us and asking these wonderful questions of Nicole and her hard research work that she's done. And we want you to remember that you can still leave your questions in the chat. We'll have them available in written format up by about next week for you to review. So, Nicole, when it comes to your next steps and what you want to do to complete your research or further the research, do you have a priority list on what you would like to accomplish?
So besides the skin and sweat, right?
Well, just your next steps, like what is your first priority on expanding the research?
Okay, so after I finish the epigenetic methylation marker for skin and sweat, I want to start researching on BMI methylation markers. So that will be how we will see different methylation for different BMI. So if you remember, BMI is the body mass index, and that is actually for underweight to all over overweight. That will actually give us a better insight on what we will get on the dimensions of the person. And also I would like to include another marker that will be for diet. Those are my next steps after I finish the skin and sweat. And of course, I skin and sweat, I still have a lot to do. But hopefully I finish by this year, if not next year, the latest at spring. Those are my goals right now. And of course, do the paper for this as soon as possible.
So, Nicole, in the last few minutes that we have for your presentation, which you have done very well, I wanted to give you an opportunity to kind of recap for anyone who is joining us and did not see the very beginning of your presentation, just to kind of add any conclusions and just do another recap, and maybe that'll ignite some more questions. And again, if you do have anything to ask Nicole, please feel free to add it to the chat. We will be saving the chats and have Nicole answer in written format so that you can get answers to anything that was asked previously before you joined us in the room, or if we run out of time to get to your question.
Okay, thank you, Michelle. So basically, my research is on the development of epigenetic methylation markers for skin and sweat. And as a recap, what we want to do is to just get some methylation markers for skin and sweat. Uh, because there is no literature on skin and sweat for epigenetic methylation, we did an Infinium human methylation 450 bit chip array. That this helped us identify different gene locations that had different CPG locations that the machine encountered different methylation percentage for the different body fluids. So for example, we did the array, and after the array, we actually discovered five different gene or CPG sites where we could see different I'm sorry for using different so many times, but we could see differences in body fluids. For example, we can see here sweat is very different or has a lower methylation than other body fluids. And our methodology is basically to collect the samples from people doing exercise. What we ask them is to just do their normal exercise and wait a little bit until they cool down. I give them the swabs, they swab their foreheads, and from there I just do the DNA extraction by using the DNEC blood and tissue kit. That's been the best extraction method that I have found for skin and sweat. Afterwards, we do the quantitation that we use the qubit. Then we do the bisulfite conversion using the Epitech fast bisulfite conversion kit, and then we do the high resolution melt. So for the high resolution melt, we will see a plot of the differential of fluorescence in function of temperature, I'm sorry, for sweat, for different body fluids. So what we want to see is that we see a different um, mm, temperature, a different melting temperature from other body fluids. We can see here that sweat, the green one, has an actual lower temperature than the other one. So it's more shifted to the left than the other body fluids. And when you plot this, just having the highest point from the just having the highest point from all of the samples, we can actually see that sweat has differences from other body fluids. For example, on the last two, those are the best ones that we can really see that specific trend. You can see that sweat has a very more lower than the other ones, has lower mean melting temperature from the other ones. So my research won't stop here. I still have a lot to do. HRM is a very good uh, technology to use, but my goal is to take this to the pyro sequencer, the PyroMark Q48 from Kaizen, and see uh, if we can actually see these results there actually. And from other research that we have performed in our lab, we think that this is possible to just see these results there in the pyromark in the pyro sequencer. And finally, I want to do the validation studies for this, such as degradation, sensitivity, and mixture resolution. If you have more questions, please feel free to ask.
We just have a few final minutes left in Nicole's presentation. So if you do have any other questions that you'd like to get answered, please feel free to put them in the chat. And again, since we are recording today's session, we will provide this on demand later on our GFJC YouTube page. Nicole will also be answering all the questions in a written format as a additional document to the viewing. So just chat us your questions. Nicole will get to the ones that we have time for. If not, thank you so much for stopping by. Nicole, thank you so much for presenting your work. And feel free to join the main webinar as we have about 7 minutes to the next presentation. I do have one question, Nicole, that we can get to before having to close out today. So are there significant sources of bias and uncertainty in your methodology as compared to other methods?
But I haven't encountered bias in my methodology. I think it's a very good one. Uh, but that's a very good question to ask. I will have to look more into it, but I think I haven't encountered any bias yet.
Okay, we are officially going to close out the chat and let everyone return to the main webinar. Again, thank you so much for taking your time and asking your very thoughtful questions for Nicole so that she could present her research. Again, thank you so much, and please enjoy the rest of the Virtual Forensic Science Symposium.